Okay. We are back.
Morning.
Yeah. I was sharing this experimental study. You know. So where I looked at the expression of toll-like receptor 2 and toll-like receptor 4 in monocyte following a stimulation by the genital secretion of HIV infected and uninfected women who were suffering with symptomatic vulvovaginal candidiasis. So you see, I extracted the monocyte because the hypothesis is that the monocytes are able to play a major role in controlling vaginal candidiasis in a woman who are suffering vaginal candidiasis. You know, especially they are HIV positive, they develop severe candidiasis infection. Now, I hypothesize this on the role of monocyte in controlling that infection. But then I know that the monocyte can control candida through two types of receptors. We have the toll-like receptor 2 and the toll-like receptor 4. So monocyte use those receptors to try to control candidiasis infection, you know. So then what I did was to try to, like in a group of women who were HIV positive and
iPhone. iPhone. No,
okay, women who are HIV positive. Can you please mute? Can you mute your mic? Can you mute your mic? Thank you. So what I did is that I have monocytes. I extracted monocyte from healthy individuals. Then I extracted monocyte from the blood, the peripheral blood. Then I know that monocyte has the two receptors against the candida that can be expressed. We have a toll-like receptor 2 and 4. Then I purposively, I can use a kit to block some a reagent to block one of the receptor and leave another receptor open. Then measure the level of cytokines. Or I can then change and block the first one, block the other one, and measure the cytokine, then block the two receptors and see what will happen. You see? So here I do it like in a group of women who are HIV positive and they are suffering from vaginal candidiasis. Then I measure the level of cytokines in their vaginal secretion, you know, after blocking toll-like receptor 2 in the monocyte. Then here I also look at those who are HIV negative, but suffering with a vaginal candidiasis. Then after blocking toll-like receptors, you see that the profile, the concentration of these cytokines is changing. If you compare this, where we have high level of interleukin 6, 10, and 13, both increased after blocking toll-like receptor 2 in HIV patient. And if you compare here in HIV negative, you will see that only interleukin 10, the level of interleukin 10, that changed the more. If you see here, after I have blocked the two receptors, 2 and 4, you will see the baseline here. So you will see that the baseline level of cytokine is almost the same like after blocking toll-like receptor 4. Then from these, I draw a conclusion that it's toll-like receptor 2, toll-like receptor 2 in monocytes that is playing a major role in controlling vaginal candidiasis infection in HIV-infected women. You see, so this is an experimental design, where as a researcher, I was able to control the situation. I was able to manipulate. I was able to, to change the concentration so that I assess the variable of interest. Is there any question about the true experimental design? But we still have other type of experimental design beside that one. Is there anyone who's study or he want to do an experimental design? I don't know what is happening here. Oh, Pam, you raised your hand?
No, for me, it's my job. I'm doing experimental design in my day-to-day work.
Oh, okay. On the RCT.
Yes, we are doing the RCT.
Okay. I don't know why my slide don't want to move. So can you see my slide?
Yes, we can.
But they are refusing to move. Okay, so we can go.
It just moved now, bro.
Yeah. So we have another experimental design that we call pre-test, post-test control group. So pre-test and post-test control. So this is also called a classic control experimental design, So here you have, we will come back to randomized control trial because that is classified among the epidemiological studies, but you will see that under epidemiological studies, we also have experimental design that we call a randomized control trial. Okay. So that's where we will talk more about RCT. Now, the pre-test, post-test control group, here you have controls. So they control the assignment of subject to experimental and control group through the use of random numbers. So actually you want to do a group, a study where you randomly, select participants. You randomly select participants and place them under either experimental group or control group. Okay. So then this procedure guarantees that all subjects, all participants have the same change of being in the experiment. They have the same chance, either. It's the same chance of being either in an experimental group or in a control group because you randomly place them. You use a random number, you place them. Okay, if you get this, you go into the experimental group. You get this number, you go into a control group. So because you randomly place them, then there is some level of control, that you control. Then because of strict random assignment of the subject, this I should not be using the word subject here. You will excuse me, because in the South African context, we, according to the National Research Ethics, we are not allowed to use the word subject. But this is mainly from American literature, where the word subject are used, but we use the word participant in South Africa. So because of the strict random assignment of the participant, it is assumed that the two groups are equivalent. So that we call also an equivalent control group design. It's also called equivalent, So equivalent on all important dimensions. And that there are no systematic differences between the two groups. So because you try to randomly select who are the participants who can be in the experimental group and those who can be in a control group. Now, since you randomly select them, then you believe that the two groups are then equivalent. They are equivalent, okay? So researchers may substitute matching for random. Then after you have randomly selected them in two groups, you also match them. You also do the matching. You match the experimental group versus the control group. So you can match them maybe according to age or to gender or to race or to their income level. You try to match them, So usually it's not easy to match more than three, using more than 3 or 4 characteristics. Usually we match the cases and the control maybe by using only 1 or 2 variables, or usually age and gender. It's very easy to match them. Then you have well-known equivalent group. Okay. So then the controls the timing of independent variables. So now the reason we call it pre-test and the post-test control group is because you then assess for the dependent variable at the beginning, so you have a baseline assessment. You have a baseline assessment, then you put the intervention, then you do also a post-test. You do also another assessment at the end. So you do an assessment at the beginning, before the intervention, then you do an intervention, then you do an assessment after the intervention. So that's why we call it pre-test, post-test control group. Now, the reason we call it a control group is because you randomly assign the participants in either experimental group or a control group, and you match those group maybe by age or by gender. So I can show you here. So you assign people in the treatment group and the control group. This is also called the experimental group or control group. Then you do a pre-test. A pre-test is a baseline assessment. Then you put an intervention. Then you do a post-test, okay, to try to see. Now the control group here did not had an intervention, but you still assess them after that to try to see if there are still any changes after a specific time period, so that's the pre-test, the post-test. I want to ask Casey, your design for your project, what do you think it is? Before you respond to that question, let me explain the post-test only control group. The post-test only is the same like a pre-test, post-test control group, but the difference is that we don't do a baseline assessment at the beginning before the intervention. So we randomly select a group and the control. We don't do a baseline assessment. We put the intervention, then we assess only post-test at the end of the intervention. Yes? Sorry?
Yes, Prof. Are you asking me?
Yes, I was asking you the design of your project.
My project is, I'm not very sure now whether it is pre-test and post-test. because this participant, it is not randomly select. It is all the students who are there. They are the part of the study. And they will do this, write this test, let me say, write this record, and then we score them, and they finally at the end of the year, they will write the final exams. And then in between, yes, there is a time, a lot of classes are going, a lot of discussion are happening, yes. Then finally they write the exam, and then we'll see, compare these results. The final one and the record one, which they write it. Is there any difference or is there any relationship in each other? That is what
you then
to do.
To think out if you are doing a pre-test, post-test control group, or you are doing post-test only, or it's none of this.
I think it looks like more post-test because they write the records. Then that can be in the beginning of the year, and at the end of the year they write the exam. Okay. And we are comparing these
doing
two
the
results.
Evaluation at the end.
Yes, so it looks like both. There is no assessment in beginning, and there is no intervention in between, and there is no test again after intervention.
Okay.
So it looks like pre and post. It looks like more post for me.
Oh, okay. Now
but I'm doing another. Yeah. I'm doing another. Can I talk another 1 minute?
Yes, yes.
Yes. I have done another small study just for my interest over the teaching methodology. I'm just trying to. I am a strong believer of active learning, not passive learning. The way of we did in our student time. The lecturer comes, give the lecture, and go, and don't care whether we understand or not. This is the more this teacher-centered learning. I am the advocate of the student learning, more engagement with the student. So let the student more be involved in the learning process. So I do these things. I used to give them to learn something, to read something themselves. I randomly select the group. In the group, I divide them in 5-6 group with 4 or 5 people in one group. That is selected randomly. And then give some materials to read, maybe half page to 1 page, give 1 hour to read and to discuss among the group. And they write a paper there, matching question. Then after I intervene, I teach them. I teach them and then let them practice again. And they talk each other, they discuss each other, and they will come and present to the whole group.
Okay.
Then again, they write the same test which was given before, the same matching. But the interesting things in the matching question, there are only 10 questions, but there are 25 answers. So some of the questions have got multiple answers. Then I just compare the results.
Okay.
And it was so interesting. For me, the group is the same people.
Mm.
But they mention the in between, they study themselves, then I come inside and teach them, and then we test again. So changing whether they can learn themselves better than without the
So
teacher,
you
or
do
they
the
still need
pre-test, the post-test.
to experiment. Yes, pre
But
and
then
post.
the problem with your group is that they are not randomly placed in different group, or it's the same group only.
No, this is the same group.
Mm.
There are for example, there are 50 students in one class. Then I randomly put them in different group. That is only randomization come only when I am putting in the group.
Okay.
Yeah.
Okay. Yeah.
And then after that group, they
Otherwise,
learn.
if you don't randomly put them in a group, we call that a non-equivalent group.
Oh, okay.
Yeah.
Then
So then you
that
can
also.
call it a non-equivalent.
Okay. Then good.
Yeah.
Thank you then.
Yeah. Then we have also what we call time series. Time series design, it's also the same thing. It's a pre-test and post-test, but instead of measuring just from the beginning and the end, we do the measurement also in between, you know? So we do several measurements. So this is a variation of a pre-test, post-test design with data collection done over regular interval of time, and the aims to assess an intervention or a program before, during, and after it's concluded, you know? So when you do that, then your design is what we call time series. It's time series design. So I can't see the hand there. If you have a question, you can just speak. There is something that went wrong with my computer. I can't see the hands anymore. Then we have another experimental design. It's also what we call factorial design. Now, factorial design are experiments where several factors are examined simultaneously. You know, you have several factors that are involved. Now, this is a study like we are doing with Urgent, is one of my PhD students. Is using a factorial design, trying to come out with a mathematical model to try to predict hypertension, you know, in HIV positive patients. So cardiovascular, the incidence of cardiovascular diseases in HIV infected patient. So he can look at different factors, you know, because there are non-modifiable traditional factors. You have modifiable traditional factors. You have HIV-related factors. You have ART-related factors, you know. So the non-modifiable factors that are associated with cardiovascular diseases like your age, gender, ethnic group. So those are like your X, then you have X1, X2, X3, then you have the modifiable factors like your cholesterol level, BMI, type 2 diabetes, and all other underlying conditions that can be associated with cardiovascular infections, you know. Then you also have HIV-related factors like your CD4 count, viral load, immune responses, and you have also antiretroviral drugs, different regimens. So when you are looking at all different factors that can influence a specific outcome, then you can use an experimental design that we call factorial design. Okay, so that's what we call factorial design. Now, let me just introduce to you non-experimental design. We might not be able to finish today because we remain with 20 minutes. It will be 4 o'clock, then we continue tomorrow. So non-experimental design are also what we call epidemiological design. So the purpose of non-experimental design is to describe phenomena and explore and explain the relationship between variables. So that's what epidemiologists mainly deal with, those type of design, you know. So here there is no manipulation of the independent variable during non-experimental design. So there is no intervention in general. But with the exception of one type of that design that we call randomized control trials, where there is it is considered as an experimental design. But I don't know why they classify it not under true experimental design, but they put it under epidemiological design. So probably the reason is that the study is conducted in a natural setting and the phenomena of interest are then observed as they occur. So this is a summary of epidemiological design. So you can see that all epidemiological design are divided in two groups. We have a descriptive study design and we have analytic study design. So most of you, the work that you will be doing probably might be found themselves here. So the descriptive study design, example of descriptive study design is a case report, case series, incidence studies, cross-sectional studies that we also call prevalence studies, and ecologic studies. So we will go through all this in details, all those descriptive studies to understand that case report, case series, incident studies, cross-sectional studies, and ecologic studies. Then we also have analytic studies. Now, analytic studies are then broken down into experimental. You see the word experimental comes here again. Now, this is an analytic experimental study. You know, it's an analytic experimental study. That's where you find your randomized control trials, clinical trials, the RCT. The clinical trials are found here. It's an analytical study. It is epidemiological study. It is analytical in nature. Then it is experimental analytical. So it's a bit different from all the other experimental that we have described before. Then we have observational study. It can be analytical study, can be experimental. It can also be observational. Now, among the observational, we have the cohort study and the case control study. So the cohort study can be prospective or retrospective, and we have the case control study. Okay. So all those are the one we are really going to discuss with more details tomorrow because we are running out of time. But before we take maybe final questions, let's just make the difference between descriptive or analytic study, because we say that epidemiological study can be descriptive or analytic. Now, the difference between a descriptive study and an analytic study, or before I respond to that, let me ask you that question. So what do you think the difference is between descriptive and analytic? Anyone? You can just speak because I don't see the hand. The difference between a descriptive study and analytic study in epidemiological design. Yeah, Casey?
Yes, sir. Thank you. Maybe descriptive is more to describe the characteristic of the study, and but the analytical is just to experimental is just to have the strictly measure this difference between this I think these variables, dependent and independent variables, and what is the causality between them. I think that could be the more analytical one. And descriptive one, the difference between them.
Anyone else? There are Yakusu. Yakusu, can you unmute yourself? Yes, go ahead. Difference between descriptive and analytic studies. We lost Yakusu. Anyone else?
Hi, Prof. I think descriptive is more qualitative and analytic is more quantitative.
Okay. You think descriptive is more qualitative, analytic. So here we are mainly talking both about the quantitative studies. Remember, we say we are going to deal with a qualitative studies later. So all those are quantitative studies. Epidemiologic studies are quantitative in nature, but then they can be descriptive or analytic. So what do you think the difference might be between descriptive and analytic? Yes, Yakusu, you are back. Unmute.
I think, Prof. I don't know if you're listening my now.
Sorry.
Okay. And I thought that descriptive study occupies prevalence.
Your connection, Yakusu, your connection is very bad. Your connection is very poor.
Risk.
Sorry? What did you say?
Is it possible? Is it possible for me to write?
Okay. Okay, you can write. Yeah, you can write. Okay.
Okay, thank you. Thank you very much.
Yeah. Anyone else? Difference between descriptive and analytic?
I was saying, Prof, I think that descriptive studies is basically trying to focus more on the hypothesis that the researcher has set out. For instance,
that one is what? Descriptive or analytic?
Pardon, Prof?
That one is the one you say you are focusing in hypothesis. Is it descriptive or analytic?
It's descriptive. I think it tries to achieve maybe like trying to answer your aims of the research and your research questions. For instance, when you're creating your hypothesis, you will use questions as in like, why is it a certain disease maybe affects a certain population? So it poses certain questions. And then on your analytical studies, I think it's more now trying to analyze why did that thing happen? Why did that disease affect a certain population? That's analytical studies. I think so.
I think you tried, but then you brought some confusion somewhere. Okay. So we will deal with this. I will read also your response, Dr. Issa Yakusu. But this is the main difference, because that's where we will start tomorrow. Descriptive or analytic. All descriptive studies are studies that will help you to generate hypotheses. Okay? Something that will help you to generate an hypothesis. It's a descriptive study. So descriptive study answers to the question what, who, where, when. You know, because you are just trying to describe. So you don't have an hypothesis. So it's amazing to see when you write your proposal, you are doing a descriptive study, but you have an hypothesis. How can you have an hypothesis in a descriptive study? In a descriptive study, you only have research questions. You don't have an hypothesis, because descriptive study, at the end of study, then it will help you to generate an hypothesis. Do you understand? And the research question that you have will be questions able to answer something like what, who, where, when. Do you understand? But analytic studies are studies that test the hypothesis. So if you have an hypothesis that you are testing, okay, you have an hypothesis that you are testing, that is an analytic study. So you are answering the question why and how. So you are then right when you say you want to know why. You want to know why, you want to know how. That is analytic. But when you just want to know what, who, where, when, what happened, when, where, who, that's descriptive. But when you say why and how, that's analytic. So analytic help you to test an hypothesis. So if your study is an analytic study, I want to see an hypothesis. I want to see an hypothesis. You can also have research question, but those questions will be why or how. They should respond to the question why or how. But if your study is going to be a descriptive study, I don't want to see an hypothesis in a descriptive study. I want to see research questions. And your research questions will be what, who, where, when. Do you understand? Okay. So we are going to start here tomorrow. Okay. Starting what time? Tomorrow we are starting 10 o'clock. So tomorrow is 10 to 12. It's 10 to 12 tomorrow. So we are going to start here. Casey, you raised your hand?
Prof, yes, I was struggling to connect. So yeah, today it was Zoom, but link was I got it the Microsoft Team. So I struggled in the beginning. So what we are going to use tomorrow, Microsoft Team or Zoom again?
No, we are using this Zoom link.
So the same link.
No, so what I did is initially I did send a Microsoft Team, but then there were some requests among some participants. Those who are outside, some are not yet fully completed their registration. So then we have to switch from Team to Zoom.
Okay, no, it's fine. I have no problem for that.
Yeah.
Is it possible for you, if you can email me also that one?
Which one? The Zoom link?
Yeah.
Okay.
You have
because
I'm using now
I send you into your WhatsApp, but I can send it via email. Yeah.
Yeah. By email and WhatsApp.
Yeah.
Thank you, Prof.
Okay. Anything else?
Thank you.
Prof,
Yes.
any chance that we could have the slides, if
Yes,
not the recording?
Yeah, you can have the slide, yeah. But
Okay, thank you
the
very much.
discussion are also being recorded. I don't think you will mind to hear your voice on YouTube.
People will enjoy it.